I believe we ended at this slide here. At a slide where I said that biological data is noisy, and this noisy level, or the level of noise, comes from 3 different levels. It comes from the level of the curation, so database annotators, for instance, not reading papers right, people trying to hide what they actually write, so database annotators not being able to read it right because it was meant to be read incorrectly, or just the fact that database annotators have to sort of translate free text into some limited number of numbers or categories. There's an experimental error, and there's a biological level of noise because proteins are not on and off switches. They work as common systems together. Prediction performance must be established properly. Sequence similarity between training and test set has to be reduced, or has to be checked, or has to be kept to a very small number. Now, did we discuss what this small number is? Did we come to that point of discussing that? So when you assess a method, you have to make sure that there's no overlap between training and testing set. No overlap essentially is defined as no pair in this redundancy reduced set having a sequence similarity, so pair AB having a sequence similarity. So the redundancy reduced subset is defined as no pair, every pair has a level of similarity below some threshold. Now what is T? What defines what T is? What is enough redundancy reduction? So let's begin with the end. Again, this is my criterion for saying that a data set is non-redundant. Can I put T equals 0? Somehow strikes me as a very safe statement, right? We have no big discussion if there's absolutely no overlap. Is that a meaningful statement?
No, because then there is probably no sequence left.
that's why. Yeah, that's a very realistic way of saying it, Daniel. Okay, it's true. I don't believe there's any sequence left. So one way of saying that is, so there's a residual overlap between any pair of sequences ultimately, because we have only 20 amino acids to give. There will be some. So if you make a very, very short alignment between, ultimately you will align to alanine somewhere or something like that. Or there are k-mers that are even longer than one, much longer than one, that will, if you have a database of 150 million, some things will have some similarity and it will not be 0. So ultimately, Daniel's point is right, you will have nothing left. So there's a practical way of answering that. I have no data left, doesn't make sense. But ultimately, there's a biological way. Biological answer is just there's a random superposition that is meaningless. Do you have a question, Elizabeth? Oh, no. So what then is my criterion for choosing a T? So we know 0 is nonsense and 100 is nonsense. We exclude, if we think about an integer, we exclude already 98. Do some more, yeah?
Can't you just look at the HSP curve and then decide?
Okay, so the HSP curve, for those of you, this is one way of doing it. So we have here the length of an alignment, typically LALI, PIDE, percentage sequence identity. The HSP curve says that if that's 100 residues aligned, and this, oh, my curve is really bad here. If in these 100 more than 33 are identical, something like that. If in 100 more than 33 are identical, or in 200 are longer than 250, that's why I need slides, is less than 20 or something like that. Everything above the curve are proteins that have similar 3D structures. Okay, that is one way of saying that. So if I want to infer from biohomology that two proteins have a similar structure, then I use the HSP curve, and I know everything above the HSP curve I can do homology-based modeling with. Okay, but I want to predict subset localization. Is that relevant now? Is that HSP curve relevant? Yeah?
I mean, isn't it independent of subset localization? Could you say you want to reduce the redundancy based on similarity, so you want to look at the similarity, and then otherwise you can use different criteria.
So ultimately your idea behind it is a very reasonable one. Unfortunately, it doesn't work. But let me rephrase the way I understand Michaela's hypothesis. The hypothesis is there is some level of sequence similarity that is defined as redundancy, and that thing is behind that curve. And behind that curve, because there is something that makes proteins randomly similar, or meaningful similar, and the difference between these two is essentially what the redundancy reduction aims at. And this is a feature that is valid for all kinds of problems. That is valid for structure prediction, that is valid for localization prediction, because it's an intrinsic feature of sequences. And there's some truth to that. But ultimately, it's not as simple as that. So we do in fact need to come up with a threshold for every single problem. And then ultimately here the T is a threshold that is specific to the classification problem that you have. You have to set it up. And one way in which you can set it up is saying, well, if A and B were such that if I know the subset localization for one, I can infer it for the other. So I can apply homology-based inference. Then that is too much redundancy. Because with one, I could provide it that homology-based inference is better than my de novo prediction method, right? Then that would be one way of doing it. So then this would be a TC homology-based inference, HPI or something like that. And now that would imply that I need this threshold between training and test set, because then I can say what I learned for the training set will predict the test set that is not handleable today by homology-based inference. Otherwise, that is the set that I know, and if I can predict a set that is within the homology-based distance of the one I know, I don't need to train and test. I can do homology-based inference, right? Essentially. That's why these two sets have to be further apart than that. Okay, so we also have an issue with sequence redundancy in public databases. Could you name some reasons why there is redundancy in public databases? As long as everybody feels you can, we're moving on. This is the kind of thing that will be asked. If anybody feels they cannot, say stop. Good. Prediction results must be interpreted by users, and we will get back to that. And benchmark of prediction methods can be difficult. Can be a difficult task. Now, if I gave you as a bachelor thesis, and I admit this is not a very exciting bachelor thesis, but if I gave you as a bachelor thesis the task to work for 3 months on showing that this statement is true, what would you do? Just to tell you one thing, I do my own assessment and it doesn't work. There's no way to prove that point, right? Or to sort of empirically prove that point. And you cannot mathematically prove that point. So it's something about empirical. What would you do?
Maybe I would incorporate different data sets and show that the methods on different data sets, sometimes one method is better than the other on this specific data set. Maybe it's worse on another data set. So that's
a great example. When you do this, you have to be a little bit careful, because you clearly can sort of manipulate data sets so that you get this result. Say we have secondary structure prediction methods, and some are better on alpha helices and some are better on beta strands, for instance. Then you just have to sort of use the different composition of the data set, and you immediately get one to win in one case and one to lose in the other, right? And that would be a relatively simple finding, in which you would not really say benchmarking is difficult, in which you would only say one is better on alpha helices and one has more, right? So you have to be a little bit careful about that. But yes, indeed, to sort of show that there are two data sets that by whatever criterion you understand, you cannot take apart. So it's not a particular feature that is better in one or the other. And yet methods are behaving differently or perform differentially on these two data sets. And you cannot say one data set is more representative than another. That would be a great example to show this. Yes? That's a great idea. Any other idea? That's in fact not what I was fishing for, but it's a great idea. It would be a very simple solution. No, not as simple, but that would be a great point, yeah. Something else? Nothing? Well, one way in which you show, so assuming that scientists are always people who try the best they can for their publications. So we're assuming a world in which people don't cheat. That may not always be true, but in this particular case, let's assume that. That means that every publication you have is the result of somebody trying to do the best they can. Now, if you went back to 100 publications and tried to use a more modern data set on the methods, on their estimates of performance, then you could simply see how often is 10 years later, with a larger data set, the estimated performance still valid, right? So it requires that you have access to 100 methods, but most likely you can easily get 100 methods for which you can get access. And most likely, for most of these, the data has become bigger and much more accurate. So you could easily sort of trace how often do people overestimate their performance. And if you see that, I don't know, 98% of the methods overestimate performance, then most likely this is not somehow maybe a social event, but most likely this is also because benchmarking is extremely difficult if nobody manages, because everybody tries to go through reviewers, and the reviewers are experts too. Again, admittedly, this is not a nice thesis at all, but this is one way in which you could do it. Okay, let's talk about a prediction method called LocTree 2. And one idea in LocTree 2 is that you have a hierarchical method. This also comes back to what LocTree did. Remember, LocTree, the original version of LocTree, essentially sort of tried to mirror the hierarchy of the natural pathway into a system of machine learning devices that, for instance, at the highest level here, distinguish between secreted and non-secreted proteins. And again, part of the reason of putting that at the higher level is because it mirrors this idea of biology. So the secretory pathway is the major path here. And this is the best decision. And since you cannot recover from mistakes you didn't do on the highest level, for levels further down, you want the best performing on the top. So these two things fit together. But what we also want to sort of add to this problem is we want to be somehow more resistant to sequencing errors. So remember the way LocTree did it, and most of the methods that we discussed so far did it, is they used some information from the entire protein. Amino acid composition, surface composition, secondary structure composition, but it was always from the entire protein, right? And that brings us back to what else could we do to have a prediction that is for the entire protein. Now, the problem is if I have a sequencing error and I cut out 20% of the protein, the amino acid composition may change, right? So, in fact, I will hopefully show you some results. I can't remember what I'm going to show you, but I may show you some results how, in fact, sensitive existing methods are to sequencing errors. We want to do it for all kingdoms. We want to include membrane proteins, which so far was ignored. And we sort of have more data. So the first question is, how can you move from this idea of using the entire protein as amino acid composition to using something else to predict a feature that still is for the entire protein? Any idea? So this exercise completely eats you up. There is no more life. How can I revive you?
Are we only talking about machine learning, or because we could use homology-based?
We will toss homology-based something into the story in the end. But we will begin with something that is not homology-based. It does not necessarily have to be machine learning. Do you have an idea? Anyone else? Or do you all have the same idea? No part of the idea. Okay. So let's just go through point at a time. And I really need my advancer for the kind of slides I have. So this is all kinds of things that we achieve with this. Let's begin with the data. So we have archaea in 3 classes, 59 sequences at the day. This is 2014 or 12 or 13. Bacteria 6 classes, 479. Eukaryotes 18 classes, 1682. So the first statement is we have really small data sets. Okay? So one way of sort of dealing with this is saying we stop and we don't do it, but we felt that we don't do that, and we try the best we can. Now, we will exclude proteins, so I'm sorry for the quality of this screen dump here. Maybe you can see what it says. So this is an extract from an old SwissProt record where subcellular localization, or location is the keyword used in SwissProt, said nucleus membrane, multipass membrane protein. And this is exactly the type of annotation we did not use, because it did not explicitly contain a tag that said obtained by experiment. Today there's actually a number coming with the evidence, and we filtered out anything that did not have an explicit mentioning to experimental. We reduce redundancy using the HSP threshold. Michaela said that already. We also use the 10-3 Psi-BLAST threshold. And then ultimately what we see in terms of reduction, and what I show here are the original numbers, then at different thresholds reduced down to numbers, and that is the 1600 that I showed you before for eukaryotes in green. Right? So initially we had 35,000, but these 35,000, applying the HSP threshold of 0, or the BLAST E value here, boiled down to something like 1600 redundancy reduced. So much, much, much smaller data set. And that ultimately is the only one we used. Then we used stratified cross-validation, where we split into 5 folds. We do 5 models. We merge those 5 models into a final model. And the question is, why do we need stratified cross-validation? So we need cross-validation. We need cross-validation in the sense where we have my data, I split it into 3, that is training, that is cross-training, for hyperparameter optimization, that is testing. What the slide before shows, now I take my training set and again split it into whatever fraction it is. So one way of doing that, into 20% black here, 80% gray, and then I rotate around and create essentially from each of these 3 splits in this model here, where I have 3 equal splits. So then since I rotate, I have 3 splits, right? So this is level 1 of cross-validation, and this is level 2 of cross-validation, where the stratified cross-validation goes onto the training set. Why would I do that on top? In fact, this is what most packets, when you use them, do for you.
I would stratify, because if you have, as we saw before, we have some kind of class imbalance, or I think also the subdivided data, like we had very few data points for bacteria and a lot more for this.
Oh, the bacteria, by the way, I'm sorry, I didn't say that. So I showed 3 solutions: archaea, bacteria, and eukaryotes. And those are different models. So those are different pathways. I will get onto that. So think about it eukaryotes only. But yes, the different classes will have different sizes. Stratified is, for instance, one way is to balance out the class distribution. That is essentially one way of doing it. Or it's one reason for doing it. Another reason. The other reason is very, very, very much the obvious. It just gives you an additional layer of, let's say, defense against over-optimization. So this allows you a little bit more to, even while you do the one training, even within that training set, within that optimization set, you still have some control over what you do. That ultimately is the answer. So because at the end of the day, that's the most likely reason. What I said, why is the correct assessment of methods so difficult? Ultimately because we always over-optimize. That gets us to the method idea. The method idea, we will use an SVM underneath. And now what we are going to use are kernels. And let's just cut to the chase and use and describe the kernel, the profile kernel, the one that we actually use. So we begin with the query sequence here, up here, and we have 3 proteins that we believe are related. In this particular case means we believe they have a similar subset localization. Okay. So then down here I turn this a 90-degree turn the information up there. So the sequence is running, the query sequence are running in that direction here. Here it's running down. In that direction, what you essentially have is the 20 amino acids. Now, what you put in here is the frequency of each amino acid. Let's begin each amino acid at every single position. Let's begin with the M. The first residue, the M, has 100% M's in the alignment. The second one has 50% K and 50% M. 50, well, it's only the M that you see in this table. The K is not written here. The third one is 50% A. Wait a minute. MKW. Why is it 50? Oh yeah, 50% A. Yes, exactly, 50% A. 25% T. Yes, that is here, and the W is missing in my my plot, and so forth and so on. This is a simple frequency, right? Trivial. Then I do a log frequency conversion. I get that table, and that log frequency conversion is shown here. I'm going to show you. This ultimately is the log frequency conversion. And now what I'm going to ask is how many k-mers are in my protein, or not how many k-mers. I'm going to sample all the k-mers, and the example here goes for k equals 3. And I'm going to look for the k-mers that are conserved. So essentially I'm asking, of all the possible k-mers in my protein, how many are conserved? I'm going to describe my protein by the conserved k-mers. That begins with the fact that I'm going to have the protein as a vector, if k is equal to 3, of 20 by the power of 3 components. So it's 8,000. I have a vector with 8,000 components. And they will be 0 or 1. 0 means I have no conserved k-mer of that type. 1 means I have a conserved k-mer of that type. The way I actually find out, and each of these positions here stands for one particular k-mer. So this 42 here is cat. The way they are ordered is completely random, but it must be sure, of course, that all vectors are ordered the same way. The way you order them, there's no information in it. It's just that they're all ordered the same way. Let's look at this cat. Yeah?
Modern question, what exactly does the term kernel mean in this case? I mean, what exactly does it do once you have it?
Essentially what you're trying to do is you're going to try to replace the SVM normal kernel function. The kernel function is the one that projects the problem into a higher dimension, and you try to have a kernel function that has a meaning in the problem that you have. In this particular case, the meaning in the situation that I have is a kernel that is related to an overlapping k-mers. So essentially the way to think about it is I assume that subset localization is determined by some local motif. I don't know the motifs, so I break up the protein into all kinds of possible motifs, and I see whether any of these motifs have a signal. And that is going to be the base of my kernel function, okay? And in the next step, I'm going to show you that essentially the way we define the kernel is we're going to vector multiply two proteins to define the similarity between two proteins. But let's go back to the way I compute whether or not that is a conserved k-mer. For the conserved k-mer of cat, let's just go for the cat here, the first three letters. Now, let's begin with cat. So cat means that I look at the value here for C, A, T. So 1 plus 1 plus 1 is here is smaller than 5. Now let's look at one where I have one mistake here, or where I have some variant. It's not cat, I'm asking what if I had an alanine at this point here in the alignment for cat. Sequence is cat. I'm still looking for the 3-mer cat, but for the A I have a 2, for the A I have this 1 again, for the T the 1 again, so I do 2 plus 1 is still smaller than 5. CQT is sort of the same story. Let's look at the CAW, is the C1, the A1 and the W, at the position T a W is a 3. So that now adds up to 5. That's above that threshold, and therefore this one is sort of ignored, meaning that essentially these are 1, the others are 1, this is not. Okay? And then you do that for, yes?
What are the positions left and right? I mean, I cut would be only the one in the middle. Oh no, it's all 3 letters.
Yeah. You essentially have all substitutions, I mean all one residue substitution. So
does the position of the vector mean the position, not the position?
No, the position is meaningless.
Okay.
The position in the vector, I mean, you choose the position, but it has no biological value. You could do it alphabetically. Let's put it like that.
So the vector's length of the number of amino acids
is 3. Yeah. No, the vector length is 20 by the power of 3, because you have 8,000 3-mers. Or it's essentially 20 by the power of k, depending on what you choose. For this example, for which k is 3, it's 20 by the power of 3 is 8,000.
One-dimensional.
Yes. It's a huge vector. I mean, it gets very quickly very large. K equals 3 is just, we actually use a much larger, not much larger, but k equals 5. I believe we use 5, so you immediately see that this is already over 1 million. Actually 3.2 million. So then the second vector is constructed the same way, and then you compute the product, the vector product over these two. And that, in fact, is exactly the kernel function. That goes into the SVM. And with that, essentially, you predict subcellular localization. So again, the philosophy of this, or the idea behind it, and this is an idea that comes from Christina Leslie and Bill Noble, they published that in 2004 for a different problem, for in fact inferring that two proteins have a similar structure, or aspects of structure. It's just that Tobias, or Tatjana and Tobias had this idea to apply it to subcellular localization. But the idea really is to describe proteins by the k-mers that they have in common, the k-mers that are conserved. Okay?
To which protein? So what's the connection between protein 1 and protein 2?
So the way to think about it now is one of those is the one that you have in the database, and one is those that you predict. So essentially you're trying to map the protein for which you do not know the subcellular localization to one of those for which you do. And that you try to do not through homology-based inference, but through an SVM kind of machine learning device. Yeah?
But there is still some kind of similarity.
There's always some similarity. That's true. And we have to, it's a very important conclusion, Michaela. So machine learning is not removing sequence similarity. Machine learning is just sort of looking at other aspects to use sequence similarity in a way that homology-based inference cannot do. Right? This could be less sequence identity. It could be distributed sequence identity. it clearly is things where you cannot use regular standard alignment methods to make this inference, and machine learning learns some other aspects. Here, it zooms into local motives. Again, this could have been done differently, yes?
Just a short question. So these vectors have the length of the proteins?
No. Again, these have the length of all possible k-mers. All possible k-mers, again for a k of equal 3, you have 20 amino acids. So you, from the 20 amino acids you can, say k equals 2. Then you have 400, right? Out of 20 amino acids you have 400 possibilities of a double letter. A duplet letter, right? A, C, A, D, and so forth. Gives you 400 combinations, because AC and CA are different in this context, right? So that's why you have 400. So far, so clear? Let's look at the. I no longer talk about triplets. I talk about doublets now. So if I have k equals 2. K equals 2 means 2 amino acids, A, C, blah, blah, blah. So with A you can do 20. With 20 amino acids you can do 20 in that direction, 20 in that direction. 20 times 20 is 400. Still not. Is there anybody on to other aspects of it? And the first one comes here, and we get, oh, that is ultimately irrelevant, so this is to comparing different kernels. The other thing that we can compare, so it's ultimately the slide before showed that this profile kernel is actually doing a good job compared to all alternatively possible kernel functions that we tried. Now, we can also cut our hierarchy in different ways. So in the simplest way here, where we don't have a hierarchy, where we essentially do 1 against all. So we have 18 classes for eukaryotes, and we do 18 different networks. 1 is for class 1 against everything else, 2 for class 2 against everything else, and so forth. We have discussed that, right? Then there is an ensemble of nested dichotomies. So that's the 1 against all. That's the clear one. The ensemble of nested dichotomies. So an ND nested dichotomy is essentially that you sort of have these 5 classes, and then you go for the first for these 1, 2, 4 on this side, for 3 and 5 here, and so forth. So you nest the way that only that classes are only at one point in the end here, and that they always that all classes, or that some subset. So class 3 cannot be here. It's completely nested. It's a complete hierarchy, right? If class 3 is here, it cannot be here. So there's no double membership. A class is always to one side of every single split. And each of those has sort of an equal probability, and then we sort of look at the entire ensemble, meaning we look at all possible dichotomies that we can have. Now you can sort of refine this by looking at all, but in the way you split the samples, you always split that there's equal data on both sides. That's another way of splitting. And then there is another way, we call it our nested dichotomy, which is when we put some prior knowledge into this, or some prior intuition into this, and say what we're going to want is something that sort of resembles the biological pathway, where we have some decision on top that is biologically reasonable. In this particular case, we also talk about membrane proteins, so on the top level we're going to decide membrane or not. This is because biologically meaningful, and because it's the most accurate distinction that we have. So everything right of it will only be membrane related, and none of the ones left of it will be in the membrane. It is slightly different, the way we have this dichotomy for prokaryotes, eukaryotes, and archaea, because they are simpler. Archaea is much smaller model. For eukaryotes, it's essentially this model that I'm showing here. Again, on the right, you have different membrane related, that is called nuclear membrane, vacuolar membrane, there's a mitochondrial membrane, there's Golgi membrane, cell membrane, ER membrane, and so forth and so on. And then there are different classes of non-membrane compartments. Okay, so then on the highest level, I can predict whether it's soluble or transmembrane. And what is this now? What do I predict? This image that I show predicts what? Ah, darn. I have to do it differently. It's already spoiled, right? Yes, it is spoiled. My example failed. You understand what I predict? No, you don't. So my example is not spoiled. So this is a visual image of a prediction. I have an alternative to predict, and this is a prediction. What is predicted here?
Transmembrane.
Yes. And it's trivial or not trivial?
Because it's a helix.
What do you mean? Oh! Oi! That is too much. And we get to tables such... Oh no, this is still assessing the different types of nested dichotomies, and ultimately what we saw is that our intuitive choice, our choice by intuition, let's call it, was as good as the best possible solution that you could get, which is why we ultimately ended up with that one, because it's less CPU than running all possible architectures. Now, let's not get into that. Let's go into the testing phase. The testing phase has now this issue of which number are you using. And that gets me to the generic reality of any kind of master thesis or bachelor thesis. You have to compile a lot of data, and it's your decision, it's your task to reduce the data that you have in front of you to by some factor of 10, 20 or 100 with respect to the one that you're actually going to show somebody else. When you go to your talk, maybe it's 1%, or when you present a paper, it's this one slide of the hundreds that you have done, right? Or the one figure of the hundreds. It's a lot of this reduction is in your hands. You still have to look at all these numbers. Some of these numbers may reveal something, and if something interesting comes up, that's what you have to show. This may still not be the final thing that you present in a paper or in a talk. That ultimately is related to what you believe is the major outcome of the thesis. The focus is your task. The focus is part of what you do in your scientific project. Now let's begin with the simple numbers here. For a two-state prediction, there ultimately are 4 numbers that you will not really understand yourself, not really show yourself, but that you should always have in some supporting online material, in some appendix, because ultimately those original numbers allow to recompile almost all the other following numbers. These numbers are in 2 states, so you can be observed to be in the localization L. And if you are also predicted in that localization, this is the number of what people typically call true positives. The word positive is meaningless. The word true essentially means there's an agreement between observed and predicted. The sort of mirror image of that, it's observed to be not in L and predicted to be not in L, so these are the true negatives. That these are negatives and that these are positives is just a label issue, right? There's nothing special about these or these. They're completely equal. It may be that you have a problem where you predict binding sites where this group is much smaller than that. It may be that you care more for the positives, but ultimately what you call positives and negatives is a label. Now, continuing in this wording here, observed in L and predicted to be not in L is a false negative and is a false positive. Now there are all kinds of numbers you can compile. So I call this typically accuracy, and increasingly I'm pushed to changing my annotation, and I'm talking about accuracy and coverage for something that people typically talk to or refer to as precision and recall. The reason, okay, let's just look at the way people typically define precision. Precision is defined as the true positives here. One more step. This table is absolutely relevant yet not useful, because if you have here 11,055 and here 150,000 or whatever the number is, these numbers to you do not say anything. What helps you to visualize those numbers are percentages. What does that actually mean? What's the fraction of correctly classified, or what's the percentage of correctly classified? And that is, for instance, done by this ratio here, true positives by true positives plus false positives. That again is what people typically refer to as precision in this one here, and what I call accuracy. Now I call it accuracy for L, and I call it accuracy for not L, so accuracy for the positives and the negatives. Ultimately, whether I look at the fraction of these that are right or the fraction of these that are right is completely symmetric. Right? There is no, this is nothing else. It's the fraction here, in my view, should not have a different name than the fraction here. In Wikipedia, they do have different names. Does anybody in the room know what the name for this one is? I happen to not know. This is why I call this both by the same name, because that I remember. Do you guys know? So again, this is precision. This value that I define here on the whiteboard is referred to as precision. What does this one refer to? Nobody knows?
Recall?
No. We get to recall on the next slide. Ultimately, recall is this, something like that. We get to that in the next slide.
Is it Tn over Tn plus Fn or Tn over...
Is this the same? You replace the P by an N. And then it's completely symmetric, right? That may very well be true. So this then would be called negative predicted value. And the fact that you don't remember, so everybody remembers precision, nobody remembers this, as if negatives would have some different reason to be, and precision and negative predicted value, if that happens to be the right one, and it may very well be true, but maybe the opposite of recall, or the analog to recall, is not the negative predicted value, it's the other one. But anyway, it's completely counterintuitive. Do use whatever you are happy with, but remember the symmetry of the problem. So by introducing completely new words, do not forget that the problem ultimately is symmetric. So the flip side of the coin is what people call recall, what I call coverage here. On this one here, it's true positive, but true positive plus false negatives. So essentially the sum over this one up there. And this essentially means at the threshold that you put, of all the ones you could have found at that threshold, how many are right? The one before means at the threshold that you put, how many of the ones that you find are actually right. That is what precision or accuracy means, and this is what coverage means. How much do you cover? Again, I just wanted to sort of put this issue of symmetry as a topic. Now the next issue is, if you want to compute two-state performance, a Q2, how would you do that from these numbers? You guys see that immediately?
True positive plus true negative.
Divided by all. Yeah, completely right. So essentially what you do is you sum the diagonal. So in this matrix, the way to think about it is that one is sort of the percentage in this direction, one is the percentage in that direction, that direction, that direction. Those are the four numbers. Those are the four numbers that have four different names, and that I just call positive and negative accuracy and positive and negative coverage, which are four percentages, right? And then comes the Q2, which essentially is the one that sort of essentially sums over all of them. And then there is one that sort of, another way of summing over all of those, or not over all of those, over two of those is the so-called F1, which typically is reserved for the positive. So F1 is typically the geometric average between precision and recall. You can also use, of course, very often, since P and N, positive and negative, is completely symmetric, there would be an F1 essentially for the negatives, which again, I believe, I'm not even sure has a name. Is again something that one sometimes has to look at. Now, this still may not be all the numbers that you can think about. What else could you think about? So in a more general system for subset localization, you would have a confusion matrix with 18 rows and 18 columns. So what else could you? You clearly ought to look at these things. The confusion table tells you whether your machine learning, for instance, is completely not predicting some class. If I told you that in almost every prediction of subset localization, some classes are not predicted, what do you believe? What will be a thing that you can say about those classes? What will these classes have in common when you look at these numbers? So I will tell you in the confusion matrix, if you look at the confusion matrix for subset localization prediction, what you will see is let's say 150, blah, 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 some numbers, and then there will be something 15, 5, 2, so something the way you absolutely don't get the diagonal. Why do you believe that will be the case typically? Or what will you, my way of asking. Okay, what I'm sorry, this is something so simple that I shouldn't ask a question because I don't know how to ask the question without confusing you. Ultimately, the answer is that the classes that you will miss are typically the ones that are very small. Machine learning almost always does better for the larger classes. The more data you present, the better it gets. That means in this confusion matrix, you see where things get stuck. And this is very important to see sometimes. Okay, now back to the next issue. The first question here, more digits imply better analysis or better work ethic. So I have two methods, AND1 and Web2. AND1, as you can see, it's a three-state performance here. As you can see, AND1 does numerically do better. And what I now claim is that it's not only doing numerically better, you see that the authors do a much more careful job in assessing it because they provide many more digits in terms of precision of the degree to which they estimate performance. Statement about that. Can you react to that?
There's no standard error reported, so you can't even tell if the digits are valid or not.
Clearly, just more digits, to assume that more digits is better is total nonsense. What you absolutely need to do is you need to provide an error estimate. And the error estimate, typically from a distribution, you have a standard deviation. From the standard deviation, you can essentially do the standard error. And the way you do it, essentially, rule of thumb, you do standard error is the sigma divided by the square root of the number of samples. In this particular case, it's plus minus 0.4. Now, if the standard error would be 0.8, how many digits should I show? It's very clear that this last thing is not supported. Nobody has a doubt about that. How many do I have to show? And again, there's no clear answer. So there are two answers that are both defendable. And in fact, essentially it depends on the way you want it. But if you said 5, then that clearly is the wrong answer. Yeah?
I think you shouldn't go any further than the number of digits that the standard error has.
This is an interesting approach, and that's a very, very good one, I believe. So if the standard error is reported to 0.8, that means you can at most, so you can cut off, or you round off, so this would be 73.8, and this would be 70.0. That ultimately is the point, yes?
But if you only have one digit after the comma, then if you have 0.8, it can still be low, so you should...
No, wait. No, but I'm not sure that I understand what you mean. So let's first get to the proposal, and then I get into what you're trying to argue. I didn't get it, I'm sorry. And now it's plus minus 0.8. And now you say what?
I would have one more digit.
Yes. So you want to put this doesn't change. Yes, it does change. And why?
Why I would put one more digit? Because it's 0.08, so it can change the law. I don't know how to explain this.
But it does make a difference.
Yeah.
It's not captured within the plus minus 1.8. Is it? It's not. With respect to the 0.8, it does not make a difference. Yes, so this turns into, if I don't do that, this turns into a 70. That is true, but on the plus minus 0.8, this still doesn't make a difference.
But if you had like a 73.2,
So you can construct an example where it makes a difference. I take that point, Vanessa. But actually, let me give you the counterpoint. In a universe where I rounded up to one digit, you cannot construct an example where it makes a difference. So if that would have been 8.5, then you could. But provided this error estimate being 0.8, you cannot construct in a universe of just one digit an example for how it would differ. And that is ultimately the argument. Michaela?
I would argue that, I mean, it does make a difference, of course, if you keep the...
Not if you, if not...
ones, but the difference is very small, and the question is like, if you really need to...
No, but again, so if let's go back to what I'm saying literally. The moment I round to this number, it does not make a difference. Okay? And that number, so if the error would have been like that, and I would have to go one more, and then it would make a difference. But then I take Michaela's point. Ultimately, what we are after is to distinguish between things that are meaningless, small differences, and those that are meaningful, or at least meaningful, not in a scientific meaningful way, but in a scientifically meaningful way. Now, let's just cut it and assume that I take the point of Michaela as given. So the limit is the limit provided by the standard error. Now, what if somebody gives the standard error like this? Let's make it a little bit more spicy. Okay, even more spicy.
Then I'll just ask the person why they...
Why do they do that? And then they say,
You just take this first.
Exactly. So the point again, so this is what I would recommend. Ultimately, this number, this value is so high that assuming that you care about zeros, any zero trailing zero after that, or even ones or twos, is irrelevant. Again, the story may slightly change if that is a 6-8. That would turn the story. And ultimately, there is no clear answer whether that is already turned with the 0.84, which you typically round down, but it is still a marginal fact. But clearly, something like this is not a marginal fact. Let's just say within a tenth of that error, this is no longer making a difference. This is the way to think about it. Okay, now